I'm Laurakay Bruhn, and I'm from Agilent Research Labs, which is the corporate research labs for Agilent Technologies. And I'm going to tell you today about some work we've been doing using, as Jacob mentioned, chemically synthesized and modified guide RNAs for CRISPR-Cas genome editing. And so I don't think I need to give any introduction at all about CRISPR to this audience. If you needed it Monday, you surely don't need it by now. So I'm just going to kind of dive right into more the chemistry and biology part of this. So chemically synthesized RNA has some advantages over making RNA by in vitro transcription. In vitro transcription is convenient. It's fabulous. The systems people have worked out in kits make it very easy to do that. There's some limitations, though, when you're using an enzyme to copy a DNA sequence into an RNA in terms of both the things like having defined ends of the sequence that you want, because some polymerases prefer, say, to have all the RNAs start with a G, and there's often kind of a heterogeneity at the 3' end. And of course, with both systems these days, you can have whatever sequence you want. There's no difference there. But getting scalable synthesis of RNA with consistent purity in well-defined amounts, especially as you get to larger scale, tends to be more straightforward with chemical synthesis. Probably one of the biggest reasons that chemical synthesis of RNA is of great interest is you can basically do, at will, syntheses where you put modifications into the RNA at whatever position you want of a very wide variety of modifications. So you can add modifications that might enhance the stability of the RNA, the specificity where the RNA is going to be hybridizing either within its own secondary structure or in the case of guide RNAs, but you can imagine making modifications on the bases that might affect the specificity of the binding of the RNA to the DNA target sequence. You can certainly impact activity of RNAs with different modifications. And you can also add modifications that allow you to do things like cross-linking and labeling, say, with fluorescent dyes. So just to kind of make sure everybody's on the same page, when we talk about chemical synthesis of RNA, it's very, people probably learned in chemistry class a long time ago, phosphoramidite chemistry that's used for DNA synthesis. And you basically start with a group on a surface, often glass or something else, and you build up through cycles of chemistry a longer and longer chain of nucleic acids, and till you have a complete RNA. And you have protecting groups on the bases and stuff so that it doesn't branch off and the chemistry all goes in the right order. And then you have to get those protecting groups off when you're done. And then there's a variety of methods for purifying the subsequent nucleic acids. So Agilent actually, quite a long time ago, started a project to try to make an improved way of doing chemical synthesis of RNA. It turns out that the little hydroxyl group on RNA, which is really the only difference between RNA and DNA, makes it much more difficult to do what I just described in terms of the chemical synthesis. And this method was developed quite a number of years ago. The original publication came out in JACS in 2011. And I'm not going to get heavily into the chemistry. By the way, I'm not a chemist. I'm a molecular biologist by training. I manage a group of chemists. I've learned a lot of chemistry from them. And I can answer questions up to a certain point, and then I'm going to refer you to my colleagues. But it's really fun and exciting that this chemistry that was developed long before anyone was thinking about what people are doing now with CRISPR and Cas9 has properties that make it able to, for example, synthesize RNAs much longer than traditional syntheses methods could easily do. So we routinely make 100, 120, we've even made 150 nucleotide long RNAs using this chemical synthesis method. And again, I'm happy to try to explain a little more about why this method works that well. And there's multiple different aspects, including the new protecting groups that were designed specifically for this chemistry, which is why it's called the tcRNA chemistry, which stands for thiocarbamate protected. The thiocarbamate is the TC part of it. Those are the protecting groups that were designed for this. So I've been with first HP Labs and then Agilent Labs for almost 19 years. And there's 2 or 3 stories I could tell about sort of serendipitous. You have an amazing new technology, and then something is discovered that makes that new technology even more useful than you ever imagined. And this is one of those examples. Because until a couple of years ago, most of the people that were most excited about being able to get chemically synthesized longer RNAs with a bunch of modifications were people doing like RNA structure function studies, doing NMR, crystallography, things like that, who also are really excited about this chemistry. But recognizing that single guide RNAs are in that 100 to 120 nucleotide range, we started making guide RNA sequences some time ago. This is an example of one of the first ones. And this is a 113 mer guide RNA of a sequence that was published a couple years ago to CLTA gene. When you work with chemists, you learn they really love mass spectrometry because they like to make something and then prove to the atom that they made what they said they were going to make. And so the mass spectrum shows on the left there is the result of such an experiment. A lot of molecular biologists are much more comfortable with this image, which is actually the gel version of a bioanalyzer trace showing the same RNA. And so I mentioned that chemical modifications is one of the things that you have a lot of flexibility to do when you're doing chemical synthesis of RNAs. So of course, there's a lot of knowledge about different types of modifications that you might make that have been accumulated from people working on siRNA and these other different types of antisense oligos. There's a huge amount of work in RNA-based therapeutics. So there's a lot of ideas about how you might modify RNAs, but in the case of single guide RNAs, not much data on what you can do to modify them and still have them be functional. So we've been doing quite a lot of work in that area at Agilent for a couple of years now. And I'm going to tell you about 3 modifications and specific placements of those that we used actually in a study with Matt Porteous's lab at Stanford, which was published online in Nature Biotechnology at the end of June. So the 3 modifications, and I apologize for any chemists in the audience about the abbreviations, because they're just really made it easier to write the paper and label figures. So the 3 modifications, the chemical structures are shown here. So 2'-O-methyl, which we just abbreviate as M, then 2'-O-methyl, 3'-phosphorothioate, which is MS, and then those 2 plus a PACE group, which is MSP. And these modifications were chosen because they're expected to affect the stability of the RNA and resistance to exonucleases, as well as they've been shown in some RNAs to have effects on the immunostimulatory properties of those nucleic acids. So we placed them in the RNAs that were used for the study I'm going to tell you about at the 5' and 3' 3-terminal nucleotides. So there's 6 modifications of those types in the different guide RNAs that I'm going to show you the data from. So it's pretty easy to test if a guide RNA that you've synthesized is active by using an in vitro cleavage assay. So you have a double-stranded piece of DNA that's made with the target sequence for the particular guide RNA you've designed. And then you can use purified Cas9 protein and the guide RNA, and then you can use a bioanalyzer or a tape station or a gel or whatever and separate the cut from the uncut DNA. And that's what this, the type of assay this data is from. And so basically you can see the mock just means it didn't get any guide RNA. And then there's really 4 flavors of RNA. There's unmodified, so just plain vanilla RNA, again made by chemical synthesis, and then the M, MS, and MSP modifications that I just described. And we used guide RNAs designed for 3 different genes, which probably you've heard already, if not here, other places about those genes. And they're all potentially very important therapeutic targets. So this is the paper that I mentioned that came out online at the end of June. And the rest of the data that I'm talking about is some of the data from this paper. It's not all of the data in this day and age of online everything and supplementary data. There's more figures in the supplement than in the paper, not uncommonly. So, but I'm going to try to just kind of give you some of the highlights. So the main take-home of the message is, the main take-home message of the paper, as you can see, is that especially in primary cells, having those modifications on the RNAs affected their activity in cells greatly. You can also see, even in K562 cell lines, which were the we used for this particular graph, that when you have unmodified and then you add the modifications, you also get enhanced activity. So what we're measuring here basically is the indel frequency is calculated by tide analysis. Does everybody kind of know? Has that been discussed here? Okay, so it's a very straightforward, fast way to estimate the frequency of indels you have. You just PCR amplify the target, do Sanger sequencing, and these very clever people figured out an algorithm which they published and made freely available on the web to basically deconvolute the traces and figure out the indel frequency. So most of the data I'm going to show you is from tide analysis. Some of it's from deeper sequencing, looking at off-target effects. The comparison here is a plasmid that's driving expression both of the Cas9 protein and the guide RNA. And then the ones with the guide RNAs made by chemical synthesis went in where the Cas9 was expressed from a plasmid in these cases. So a very similar type of result was seen when, instead of just looking at the indel frequency, the Porteous lab had already set up GFP encoding donor plasmids made for each of the, with flanking sequences for each of these 3 genes. And you can just count the GFP positive cells and see a similar type of effect. So in K562 cells, a lot of people have done this. The plasmid does work really well. Putting in the guide RNA as chemically synthesized RNA also works very well. And as Jacob mentioned, you can also put the RNA protein complex in, and that works really well. So we also looked at the use of the chemically synthesized guide RNAs together with a messenger RNA expressing Cas9, as well as some studies with the Cas9 protein. So this shows basically, and here the data is presented a little bit differently. So the different color bars correspond to the either unmodified or modified guide RNAs. And again, this is K562 cells, where when you deliver the Cas9 on an mRNA, you see a similar benefit of having the modifications on the guide RNAs. So the leading hypothesis for why the modified guide RNAs work better is because it stabilizes them to exonucleases. So to try to understand a little better whether that was a factor in what was happening, we did a time course where we delivered the, so K562 cells are very robust. You can serially electroporate them and they're just fine with it. You can't do that with primary cells, but you can do it with these cells. So the data I showed you in the previous slide was the co-delivery. And then these 2 data points are where you put the mRNA for Cas9 protein in first, and then you add the guide RNA either 4 or 8 hours later. And you can see when you have the protein already in the cells, there's not a lot of benefit of the modifications, but the chemically synthesized guide RNAs still yield very high indel frequencies. If you put the guide RNA in first, and then either 4, 8, 12, or 24 hours later, add the Cas9 expressing mRNA, then you see a huge impact of the modifications. So this kind of supports the hypothesis that the modifications are helping to stabilize the guide RNAs. And also it supports the idea that if you have the protein there, or perhaps pre-complexed with the guide RNA, there's some stabilization of the RNA already imparted by having the protein there. So this is a little bit of a complicated slide, but just focus up here first. So I mentioned we also tested the chemically modified guide RNAs together with the protein. So this shows just the MS modification versus unmodified guide RNA. The yellow is Cas9 expressed from a plasmid. The green is Cas9 expressed from an mRNA. And then the red is the Cas9 protein pre-complexed with the guide RNA before it gets transfected into the cells. I think I forgot to say, all of this data is from doing electroporation or nucleofection, as Lonza calls it when you do it with their machine. And what you can see is that in all cases, the modifications helped, although the difference when you have the protein in the impact of the modifications is lower than with the other systems. There's also, so these are 3 bioinformatically predicted off-target sites, so in this data we did deep sequencing of these 3 sites. And the other thing that's kind of interesting that you can see is, well, first of all, the modifications on the guide RNAs, and there's a lot of other off-target data in the paper. This is the IL-2RG locus from also the HBB and CCR5 locus. So the main take-home message is the modified guide RNAs are not hugely different in their off-target cleavage compared to the plain vanilla RNA. There are some differences. In some cases, when you take the ratio of off to on target, it's better with the modified guide RNAs, and in other cases, it's a little bit worse with the modified guide RNAs. So I think you also may have heard a lot at this conference, as I've heard at other conferences, that there can be a lot of variability between different guide RNA sequences, different targets, and so on. So we probably need to do some more studies to understand if there are any differences when you have the modifications in terms of the specificity, or these types of modifications anyway. But what does seem pretty clear, this is a log plot of the same data, but if you can read these numbers here, when you have the protein, that had the lowest off-target activity at this particular site compared to the mRNA or the plasmid-driven Cas9. So we found that kind of interesting, and we're trying to follow up and see if that tends to be reproducible. So I mentioned at the beginning that kind of the main take-home message of the paper was about how the modifications on the guide RNAs helped a lot in primary cells. So there were two types of primary cells that we used. One was primary human T cells. The Porteous Lab has done work for years on research aiming towards gene therapy, using lots of different gene editing technologies, including the most recent popular ones, zinc fingers and talons, and now CRISPR. So they're pretty adept at doing this kind of stuff. But basically, again, this is electroporation. This is in T cells. And with the Cas9 coming off that mRNA, the unmodified guide RNA, or even with the 2'-O-methyl modifications, there's no detectable activity. But when you have the higher levels of modification, you see quite nice frequency of indels. And in all of the data I'm showing you with the primary cells, they've worked out the electroporation conditions to have like 90–95% efficiency in getting material into the cell as measured by a GFP plasmid. So there's no selection of GFP positive cells and then taking this data. This is all the cells that were in the electroporation mix. So you see, as I mentioned before, some impact when you put the guide RNAs in with the protein, but it's less of a difference because even with unmodified, you have some activity when you put it in with the protein. So these are CD34 positive hematopoietic stem and progenitor cells, again, taken from a variety of different donors. And for the IL-2RG and HBB genes, which makes sense to target in these cells, you see a very similar result as you did for CCR5 in the T cells. One thing that I think a lot of people who've been doing this kind of work know is that putting DNA into primary cells can have some deleterious effects on the cells. It can be quite toxic. And there's a fair amount of data in the paper, especially in the supplement, looking at different characteristics of the T cells and stem cells to make sure that they're all okay. I am just showing this one example just to kind of hammer home the point that having an all-RNA or an RNP system can have advantages just in terms of not having as much toxicity to the cells as what can happen in primary cells when you try to put DNA in. And so this shows basically that the percent of cells that are dead 3 days after the initial transfection, and you can see that when you have the plasmid expressing Cas9, it's significantly higher. So as I mentioned, there's a lot of other data in the paper, and but those are sort of the highlights. So just to summarize, what this data suggests is that certainly modifications that would be anticipated to affect stability to exonucleases do seem to have a beneficial effect, at least in some cell types. And as I said, we're continuing to try to further understand where you can modify guide RNAs and still have them be active, and different types of modifications to affect different properties of the guide RNAs. And I mentioned before that another advantage of doing chemical synthesis is you can get RNA made at different scales. And so I show here kind of, a few RNA made in tubes. Of course, you can make plates of RNAs, and we're scaling up our RNA production, the research scale RNA production to do this. You can also, how many of you have seen a kilogram of RNA in your life before? Yeah, so I have. And the reason I have is because, and this is a picture of a gentleman standing next to about a kilogram of RNA. And so probably not many people have seen a kilogram, and probably not many people know why I've seen one. And it's because Agilent bought, actually, several years ago, a company that makes they manufacture oligonucleotide active pharmaceutical ingredients under contract to various pharma and biotech companies. And so that's where that kilogram of RNA came from. It was pretty quickly, of course, packaged in a different way than just in those plastic containers before it got further processed. But the site, the company is actually in Boulder, Colorado, so that's why the nice mountains back there. And this company has been doing this kind of stuff and supplying things worldwide for over 10 years. They're very good at it. And so the kind of exciting thing from our perspective is, of course, with CRISPR technology having a lot of promise for therapeutics, we can go with very compatible chemistry from small research scale all the way up to this kind of scale. If you try to do this work and switch from one chemistry to another, as you go from small scale to large scale, as you get into clinical trials and stuff like that, it probably delays things by about a year because you have to redo a lot of stuff. So that's just a kind of note about large-scale synthesis. So just to close out here, I just want to acknowledge there's quite a number of people from Agilent who have contributed to this work. And then we've had a really fun and productive collaboration with Matt Porteous and people in his lab. We like to do science that will be helpful in terms of new technologies, new measurement technologies, potentially clinical relevance, but we also like to have fun doing it. So and I've listed my email address here in case people have questions that they want to follow up on.